Good morning, everyone. So thank you for waiting. We just had some connectivity issues this morning with one of our speakers, but they are now online, so good news. So first off, just a thank you to everyone for joining today's webinar. My name is Brett Verdeman, and I'm part of the MGI Africa and Europe team. And I'm very happy today to welcome you to today's webinar. So we are very pleased to have Prof. Iwalokun and Dr. James from the Nigerian Institute of Medical Research to talk about high-throughput sequencing of SARS-CoV-2 complete genomes from respiratory samples using MGI's DNBSEQ-G50, a Nigerian experience and lessons learned. So while all of you would have seen the abstract while registering online, Prof and his team have sequenced hundreds of SARS-CoV-2 genomes using the DNBSEQ-G50 to contribute to informing a robust and dynamic public health response for the containment of the ongoing COVID-19 pandemic in Nigeria. NIMR is also supporting the country's public health authority in the genomic sequencing of infectious strains, providing knowledge and support locally and to other countries within the continent. So very cool. So the speakers today will be Prof. Iwalokun, who is the director of the Central Research Laboratory at the Nigerian Institute of Medical Research in Lagos, Nigeria. So Prof joined NIMR in 2008 as a senior research fellow. He became Deputy Director of Research in 2016 and Director of the Central Research Lab in 2020. He holds a PhD in biochemistry from the College of Medicine, has 9 research grants and over 120 scientific publications to his credit. His area of research is microbial science and biochemistry using molecular biology techniques and genome sequencing approaches. He has lecturing, research, and mentoring of postgraduates and postdoctoral students experience spanning over 25 years across 4 universities and the NIMR. He has an extensive background in biochemistry, genetics, genomics, and bioinformatics, and has led the Nigerian Institute of Medical Research effort in the sequencing of hundreds of SARS-CoV-2 genomes. So also joining us today is Dr. James, who's a molecular biologist and research fellow at the Nigerian Institute of Medical Research and he has a PhD in biochemistry and has experience in lecturing and the development of assays for the detection of genetic mutations and gene expression profiles of various genes implicated in pathological conditions. He also is a co-founder of a biotech company called GeneLab Biosciences and is currently using bioinformatics and nucleic acids to develop a bedside point of care device for the differential detection of prostate cancer in his research. So with that, I thank you again for joining, and I welcome the NIMR team to share their knowledge and experience today. So Prof, you may share your slides. Cool, Prof, we can see your screen.
on the existing protocol. Yeah. So my presentation is titled High-throughput sequencing of SARS-CoV-2, complete genomes from respiratory samples using the DNBSEQ-G50 to share our experience and the lessons we have learned so far. So this is going to be our presentation outline. And the basis of this presentation is actually to share our experience regarding the use of DNBSEQ-G50 in the area of governance, validation, and training. The workflow itself involving ATOPlex-based library preparation, sequencing, bioinformatics, data communication, and sharing of genome data sets with the global community, the publications, and the lessons we have learned. So what I want to say first is that Nigeria has experienced a series of epidemics in the last 10 years, starting with Ebola 2014. We have persistence of Lassa fever outbreak. Then COVID-19 came in on the 27th of February. So on the last count, we currently have 3,637 active cases, confirmed cases, death of 3,144 people, giving us a case fatality rate of 1.22%. This is very similar to global situation with a case fatality rate of 1.12%, but a little bit lower than what we are having in Africa as a whole generally, with a case fatality rate of 1.99%. The approach of surveillance of COVID-19 cases in Nigeria was through the framework established for Ebola, with an addition of a new data collection system called SOMAS, meaning surveillance, outbreak response, and management system. And what SOMAS entails in the early stage of COVID-19, particularly in the first and the second clusters, was actually to diagnose using any of the RT-PCR kits that have been validated, including the BGI targeting the ORF1AB, the TIB mold biotes targeting the RDRP, the GenSeq, Life River, ORF1AB, N and envelope gene, Virtual, and the one that we developed in NIMA called SCODA, which is an ORF1AB targeted RT-PCR. So Nigeria fared well at this level, but we lacked the capacity for genome sequencing of SARS-CoV-2 at the level of the first and second cluster. So what SOMAS was providing has been summarized on this slide. We have number of cases, the test positivity rate, the case fatality rate, age distribution by COVID-19 cases by age, then mortality by age. So no genome sequencing data. So like in many African countries, Nigeria too was not really well prepared for genomic epidemiology study and analysis of SARS-CoV-2. Like I said, limited genome sequencing capacity in country. SOMAS lacked the tools for collecting and analyzing SARS-CoV-2 genome sequence data. So we have limited genome sequencing infrastructure in country, and we did not have SARS-CoV-2 variant information as at that time. So pathogen genomics expert in country was also limited. And above all, there was total lack of knowledge regarding MGI sequencing platforms and its applicability. So this is just an outlay of the applicability of MGI sequencing when we started using the DNBSEQ-G50. We now know what we can really apply it for. The simplicity of the library preparation, the sequencing itself, and the bioinformatics analysis. So we now know that with DNBSEQ, we could use it for many applications, vis-a-vis surveillance, variant analysis, evolutionary analysis of SARS-CoV-2. And the advantage is that the library and the sequencing platform could offer large coverage of SARS-CoV-2 genome. The ATOPlex has a simple operation module with a reverse transcriptase reaction coupled with two-step PCR with limited steps in cleanup, depletion of ribosomal RNA, which could cause bias in reads at the end of the day. So we were trained on this. And NIMA is also playing a very crucial role in the control of COVID-19 pandemic in country. By way of introduction, is the Nigeria foremost institute of medical research. We are mandated to conduct medical research in Nigeria to improve public health and national development in the area of pathogens of public health importance and epidemic and pandemic-prone pathogens like SARS-CoV-2. We are an agency under the Federal Ministry of Health. So this is what we have at the beginning. So we have many research laboratories with facilities like thermocycler. We have genetic analyzer located in the Center for Center for Human Virology and Genomics. We also have SEC Studio located at the Central Research Laboratory, multiple thermocyclers and so. So with this, we can do gene sequencing, but not genome. So we cannot embark on genomic epidemiology study. So this is where we were before COVID-19 pandemic came in. So, and these are the research capabilities of NIMA at a glance. In the area of diagnosis, monitoring of resistance, research innovation that usually lead to the development of diagnostic test kits, research communication, surveillance and outbreak investigation, ability to do transmission patterns using PCR and Sanger. So we are limited. So this is just the platform of Ebola that was leveraged on in COVID-19 response in Nigeria, with NIMA playing a very relevant public health and research roles. So what are we looking at this presentation? We would like to describe our preparedness, how we responded to the index case, and what we did with the first 26 COVID-19 positive samples that covered the first and second waves, and also to share the lessons from our from the experience that we gain from these efforts, and also to discuss the next step. So in terms of preparedness, under the leadership of the current Director General of our institute, in person of Professor Babatunde Lawal Salako, with the support of the management, 3 research fellows were actually sent to China CDC to build their capacity in genome sequencing using the MDI platform, including my humble self. Next, we also have another set of researchers sent to Louis Pasteur, Senegal for training using another platform. Now, the Central Research Lab that I currently held was revitalized under this management. Then we acquired the DNBSEQ-G50 and MSP100 with the establishment of a sequencing workflow. At this acquisition, we were trained by Dr. Pasha and Dr. Pablo from Spain. Then the lab was commissioned by Honorable Minister of Health for State, and the ATOPlex kit was also used to validate our library prep and to train members of staff of CRL. Then we started implementing sequencing runs on DNBSEQ-G50, and we started maintaining it. What is crucial at this point, like I said, is that Nigeria as a whole lacked experts in pathogen genomics. But with the coming in of DNBSEQ, funded through our COVID-19 fund by Federal Ministry of Health, the prayer and the support of the management, and also the linkage effort of Peter and Jane, we got the DNBSEQ-G50. We were able to train personnel, and they became initially emergency pathogen genomics, but they have gone beyond being a novice to become intermediate stroke to advanced pathogen genomics in the country. So this is the process of our operation using the DNBSEQ. So NIMA has a drive-through COVID-19 testing platform, as well as other laboratory networks in Lagos and Southwest Nigeria, through which we collect diagnostic samples. Most of them are respiratory, including the nasopharyngeal swab, the oropharyngeal or the throat swab, and the nasal swab. So personnel were trained in sample collection, and we were using basically 4 validated RT-PCR, because we could have shortages in one of them, then we switch to the other. They have all been validated and approved by the Medical Laboratory Science Council of Nigeria, in which I also participated in that validation. So we will do that, and those that are COVID-19 positive, we will isolate the RNA. We will do purification of this RNA. The diagnostic sensitive value will be recorded, and then we now go on using the ATOPlex kit to generate the library. We pull, we clean using a third-party SPRI bead system. Then we pull. We do quality check of that pooling, and the benchmark was actually 48 microliter or 400 nanograms total quantity of RNA. That will be our starting material and pooling. followed by denaturation, circularization, synthesis of DNA nanoball, QC at that level, balancing of the DNA nanoball, and loading onto flow cells in our DNBSEQ-G50. So this is just the outlay of the central sequencing lab where this operation is taking place. So access to central research lab is now well organized after the acquisition of DNBSEQ-G50 for compliance with biosafety and biosecurity guidelines of the country. And we are constantly being monitored by Federal Ministry of Health and personnel from NCDC. So how did we respond to the index case of 27 February 2020? And thereafter. So at this point, because we actually acquired the DNBSEQ in May 2021. So we have had an index case, our index case in 2020. So we had challenges in procurement and getting the machine in country. And we had to wait to be trained and for the machine to be validated. So what did we do as a response, as NIMA's response to the index case? So we used our 2 genetic analyzers. We used cocktail of primers, but eventually it's not the active type, but it's primers that we, researchers in NIMA quickly designed, and we procure it from Incaba, and we use them for the purpose of characterizing the diagnostic SARS-CoV-2 in the diagnostic sample using the genetic analyzer. So when we got the consensus sequence from the 2 machines and we blasted it, we found out that what we got was an RNA-dependent RNA polymerase representing the non-structural protein 12 within the ORF1ab gene of the virus, and with 99.5% or so of genetic similarity with the Wuhan strain. And that confirmed that it was actually an index case. So the sequence data was submitted 2 days. Within 3 days, the index was reported on. So that is about actually on the 29th of February, NIMA submitted the index case. So we did by phylogeny to understand how close the index case SARS-CoV-2 variant, which is actually a B.1, to some of the reported coronaviruses in country, in Zaria, Camel Mars also, and the Wuhan strain itself, so very close. So we quickly conducted a national training of frontline health workers involved in COVID-19 diagnosis using our genetic analyzer and SEQ studio. Still, we are waiting for the DNBSEQ. Then we quickly established the drive-through, like I said, for us to organize systematically the way we collect samples, store samples, and to be able to go back and do retrospective studies. And one of the importance of that is that NIMA is also using it to monitor trends of COVID-19 cases, to be able to predict when we are going to have the next cluster. So after the acquisition of our DNBSEQ-G50 and the validation of the machine and training, we collected diagnostic samples carrying high viral load, that is CT less than 30, 26 of them, and we used it on, we sequenced them using it on the flow cell. So the first set of flow cells were actually given to us by MGI, and that facilitated the sequencing. So we followed the process. We established a protocol. So one of the challenges we had earlier after training was to adapt the protocol of MGI to situation in country, and was further sent to MGI for validation and approval. So we are able to adopt that protocol, modify it, develop our own. It's currently what we use in country. And we have also shared it with a partner, a collaborating agency that is also using the DNBSEQ-G400. We got G50, so we have another institution that is using G400. So this is the protocol workflow, and it has worked for us in that. The bioinformatics aspect will be spoken of by Dr. James. So I'm going to skip these slides and move on. So most times the ethical approval is usually waived because we are focusing on the genetics of SARS-CoV-2. We are de-identifying samples, but through our work drive-through system, we are obtaining informed consent from patients, and we are conducting in accordance with Declaration of Helsinki and the national guidelines and policy led by the Presidential Task Force NCDC. So in the area of data sharing and communication, we are reporting timely to the Federal Ministry of Health as well as NCDC, and we are submitting our sequence data to DSEQ. We are creating awareness through communication programs on TV, being done mostly by our Director General and any other person given the approval to do so. Then we are also publishing. So this is just the summary of the result that we got with the 26 samples. So we saw that those samples came up with different viral load because we use quantitative RT-PCR. And then the CT actually ranged from 15.9 to 24.8. So they are all high viral load samples because the CT is less than 30. Then one important thing is the quality score generated from DNBSEQ on those 26 samples. More than 80% of the rates had a quality score greater than 30. That's Q score of 30. We have more than 80% of the rate having it, as shown in the first Q mean quality score figure. Then another thing we found was inverse relationship between CT and the number of reads generated by the DNBSEQ on the 26 COVID-19 samples. What is more revealing is that a total of 2,093, 258, and then 20 million raw reads were mapped, and they represented with a mapping rate of 99.17 to 99.91 relative to the reference SARS-CoV-2 G1, the Wuhan NCO 45512.2. Another important thing in the first and the second cluster of those 26 COVID-19 samples, we didn't see a variant of concern. That's the first one, but we have seen a lot of them thereafter. So we didn't see the UK strain, we didn't see the Delta strain and other ones. But we saw some strains, and then some of them became the so-called the Nigerian strains. We have the L.3, we have this B.1.325. So these are the strains that we saw in the 26 COVID-19 samples using publicly shared web tools like Pangolin, Nextclade, and Gplate. So, but this is the Pangolin information for those ones. But the results at a glance showed huge diversity of SARS-CoV-2 variants that were introduced into Nigeria between March and December 2020. More of interest is that as early as these first and second clusters, there are some mutations that have emerged among these strains and have become dominant in terms of frequency. So we have the one for associated with RDRP, which is the P323 to L mutation, which is representing C to T substitution change in the region of 14,407. So that is the non-structural protein 12. Another one that occurred in the spike protein that we found in high frequency of 73.1 among those samples was the D to G amino acid change on codon 614 of spike. So this has also given us evidence that there is a likelihood that most of the introduced variants were not from China, because China variants started with the S-clade having the D614, the wild type. Or rather, they are from Europe, USA, and UK due to the G-clade dominance. So in the area of data sharing, at this time, we were able to submit 7 sequencing reports to Federal Ministry of Health. We participated in over 20 awareness programs on TV and radio stations. Most of our high-quality COVID-19 samples were submitted to GSAID. Overall, we now have over 600, more than that, submitted to GSAID. Then we have over 50 scientific papers of complex, but I must say that papers on genome sequencing are limited, but we have a lot of manuscripts already prepared for submission now. And we acquired mean iron, first through collaboration with MRC, but that gave us opportunity to sequence more genomes with the use of DNBSEQ-G50. But now that we have more of ATOPlex kits, we are now coming up with sequencing of the Omicron variants in country. So these are our submitted sequences for the first wave, for the second wave, for the third wave, and for the fourth wave. These are publications so far. So another good thing was that we were able to publish the ENAP notes describing how reliable the ATOPlex kit is and the DNBSEQ-G50 sequencer that we acquired from MGI. So all the sequences generated were able to submit them to GSAID. So one of the lessons we learned was good governance with the support of our current DG, with the support of the Minister of Health for State, And another one was also the buy-in of Federal Ministry of Health and opportunity given to NIMA to be part of pathogen SARS-CoV-2 Genome Consortium in country. We are constantly maintaining our DNBSEQ-G50 by our project staff. They were also trained by Pablo and Dr. Pasha. So we have put to use our MSP 100, even though we are still planning to use it more to automate our library prep, but we are preparing the library prep also manually. So, so we are having our regular meetings. So capacity building is what we do almost every time now on monthly basis with members of staff of CRM. We are also having constant support by our vendor, formidable vendor, Peter and Jane. So we've used this support. We have leveraged on this to establish collaboration with many international organizations like MRC, who are also sharing data from the West African region with NIMA. So we are embarking more into omics research and grantmanship. And then these are also keying into some of the products we are developing to generate the starting materials for our library prep and sequencing. But we need a big data storage facility. We need more bioinformaticians and data science experts. So in conclusion, we can say that even as far back as December 2020, Nigeria has had a fair share of SARS-CoV-2 variants being introduced into the country. And as at that time, there was IO selection for mutations that actually supported rapid viral evolution in country. So most of the samples that we got elicited variation in various logs with inverse correlation with the number of reads generated by the sequencer. But overall, we can confidently say that the DNBSEQ-G50 is a reliable sequencer to generate high quality SARS-CoV-2 sequence data for global data sharing and communication through GISAID and GEMBA. So what is the next step? So we've signed an MOU with MGI, and through that we've been giving ATOPlex kits to be able to sequence 100,000 samples, which we have started. And this will enable us to accelerate the number of sequence data we'll be generating, particularly now that we have Omicron subvariants BA4 and BA5 circulating in Europe and being introduced in African countries. This is going to give us an opportunity to investigate breakthrough infections, since we are also engaging in some COVID-19 vaccine trials. So we are very thankful, we are very grateful for the opportunities given to NIMA, to NIMA CRL, for the opportunity of training, for the opportunity of technical support, opportunity of checking on us constantly. Then we also had bioinformatics support from MDI. At one time, there was an issue of demultiplexing that was solved through a form of remote that was solved remotely. So at one time, our flow cells were replaced when the first servicing was done, and with the the ATOPlex kits that are given to us. So this is our team. My humble self, Dr. Toye, the lab manager, Dr. Fowora, molecular diagnostics, Dr. Okura Iwe in charge of confirmation of diagnostic samples, repeating the RT-PCR. Dr. C.K. Owama, Dr. James, they are also leading the role in sequencing operations and also training of the project staff. We have 8 project staff. Dr. Ajibaye, and of course, we have 2 scientific officers, J.B. Ayola and Mr. Yusuf. Thank you very much. Hello.
Thank you, Prof, for that presentation. Dr. James, will you be sharing your screen to talk a bit more about the bioinformatics?
Okay, sure, I will do that shortly.
Wonderful, thank you. And then we'll take questions after your presentation.
Sir? Ben, sir. Okay. So thank you. Can you see my screen?
Yes, we can see and hear you clearly. Thank you.
Oh, great. So Prof has done a great job in providing a detailed description of what our experience has been here in NIMA. So for the bioinformatics part, it's going to be very short, just to describe how we were able to analyze our samples quickly using the SARS-CoV-2 multi-PCR version 1.0, which helped us to generate all the consensus FASTA sequence that we needed to do our lineage assignment and do some other further analysis that we might, that we actually would wanted to do. So we got this from GitHub. So we got the information from the GitHub, and the introduction to the pipeline, an explanation on how to use the tool was very clear. So at the end of the day, we had to use the Docker version of the software, which had all the dependencies, coupled together in a single container. So it was very easy for us to run the analysis quickly. So we followed the simple steps in analyzing the data. So what we did first was to, after generating the FASTQ data for the paired end reads, we took the paired reads FASTQ data into each folder, and we named each folder as following the barcode IDs. Then we went further to prepare the input JSON file and the sample list files. So you can find these 2 files in the config folder when you download the Docker tool for analyzing this data. Then we went into the Docker environment. You might need to just do a crash course on maybe 15 minutes training on how to use the Docker environment, which could be very helpful in understanding how to analyze the data. And once you set up the analysis, it runs all by itself, and it generates the FASTA files, or FASTA files, as commonly called by some persons. So, so this is what we did. So we created all the sample files representing the barcodes for each sample. And in each sample folder, rather, we have the paired reads of the FASTQ files. So I'm using sample 1 as an example with the barcode 1 label. And this is where to find the JSON file and the sample file to edit, because it's important to go to the config folder. So once you open the config folder, you would find the sample file, which you can edit. So one important thing is that you need to edit your sample list as your as your files are being generated from the machine. So in your first column, you have the flow cell information, lane 1, and you have some of the information of the samples and the barcode ID 16, and we created a sample folder 16, which held information for the 2 FASTQ files generated with this label. And so we went into the Docker file, and so I've summarized the steps we used in running the Docker environment. So the first step is you entered your Docker environment. For me, I had to use a sudo docker run it, and I had the path for where I kept my data, which aligns with the root data information within the container, and the path for the config, and also the path for which the results will be generated. And and the script that runs the program, which it's also here. So in just running the analysis, you just, after starting or entering your Docker environment, just to run the analysis, I just typed this line, python/root repo SARS-CoV-2 multi, as explained in the instruction of gotten from GitHub. And you can cd into your results folder in root, and you can always monitor the run using no-hop sh script and see how well your run is going. So once your run is ready, right, you can open the folder, your result folder. You will see some folders that have been generated for the results. And in generating my consensus file, which I needed because we're working with a lot of files, I used a script, or we use a script find to look for all the consensus file, consensus FA files for our samples, and all put them into a single consensus file. And with that, consensus folder, we cut all the consensus files into one file, and having that single file, you can pango your results immediately using the pango script, and you can get the lineages of what we're working with. So with this, we were able to analyze our data in less than 24 hours, and this tool was really helpful and really saved us a lot of time in generating our data quickly. So one good thing about the software or the tool is I generate an analysis report that gives us information on the sample ID, the Q30 of the raw reads, the GC content, the amount of the raw reads. We can see that for one sample, we're getting over 5 million raw reads for one sample. And after cleaning the reads, we still had over 5 million. So having a clean rate of about 99.38% and a very high mapping rate of about 98.97%. So this helped us, and Prof had given a lot of information about our sample IDs and the amount of the quality raw reads that we're able to generate, very high quality, and some of the lineages we're able to generate during this run. And I think with that, I'd like to stop here, and thank you for listening and take questions. Thank you.
Okay, well, thank you, Dr. James, for sharing your presentation. So just from my side, or from our side, I would like to thank you very much to the both of you for sharing today. I think it's great to see what your lab has achieved, and also the potential for future research that will be ongoing, that will be valuable for the African continent and the scientific community. So if I look at some questions, so we have 2 questions. So the first one is, how has the knowledge and experience obtained from the COVID-19 pandemic assisted the lab with tackling future outbreaks?
Okay, okay, thank you very much. So with this experience, we have like a template that can be applied to future outbreaks. Prior to SARS-CoV-2, we had very few labs that had NGS platform or NGS pipelines. And with this, the lab is well prepared for other outbreaks. And this also helped us to train quite a number of young persons who are very, very active. Like most of the analysis you saw on our slides were actually done by quite a number of very brilliant graduate interns in our lab, you know, who handled from sample handling to the bioinformatics aspect of the work. So that makes us prepared, and that has made us to be able to, you know, amplify our efforts in terms of strength of personnel that we have, strength of people with technical ability that can help us tackle other outbreaks. Thank you.
Great, thank you. We have some more questions. Eleanor has asked, she may have missed this info, but what do you use as controls?
So for our control, we are using synthetic RNA of SARS-CoV-2, and also after a series of sequencing, some of the validated sequence data through sharing with other sequencing platform in country, like Redeemer University, African Center of Excellence in infectious disease research, and Lagos State Biobank, and 54 Gene. So those validated were subsequently used as our positive control. Then our negative control is just the buffer system. But what we also do is that at every sequencing, we balance our DNA nanoballs in ratio 3:1 with the balancing DNA nanoball that came with our sequencing kits. So that's how we have been able to do the quality control aspect of our sequencing. So we have a lot, we do external quality control with other genomic labs, very few. They also send samples to us to confirm. And any ambiguity are also done by third party. So that's why we've been doing our control. Because in the early stage of COVID-19 pandemic, there was biosafety issue utilizing live SARS-CoV-2 or getting it across countries. So we started with synthetic RNA of SARS-CoV-2, then eventually did internal validation and exchange of samples with other genome sequencing laboratories in country. I hope I've been able to answer that question, right? Are you okay, satisfied with that? Hello?
Sure. Yes, yeah, thank you. She has confirmed. We have another question from Jose. Could you tell me what platform of bioinformatics you used for the study?
So for the platform, as in the operating system. So for the we use the Docker environment, which can work on any operating system, using the pipeline developed by MGI, deposited on GitHub for analyzing SARS-CoV-2 sequences. So it's a very easy pipeline to use, and it has well-detailed instructions on the GitHub page for MGI. And so you can deploy the Docker system on any OS that you have. and have the analysis done. But for us, we use Unix platform for the analysis. Thank you.
Okay, thank you. And we have another question from Piani, who asked, how do you deal with patient information or the integrity of your data from, let's say, external parties if the database is external? Or is this not a factor in your case?
So one of the ways that we've been able to test the validity of our sequence data is through submission, because there are some strict guidelines regarding the quality of your data in terms of length, in terms of the number of ends. So, patient's data. Patient's data that we got is, we said it earlier on that we get their informed consent, 1, 2. We have these guidelines that we should focus on the SARS-CoV-2 genomics. So we have ethical waiver for sequencing. When it comes to utilizing the metadata, we get patient consent. And we use limited metadata for submission. So that is how we've been able to do that. But at the point of using the metadata, we will definitely get an ethical approval. And then that is what we are doing when it comes to writing a manuscript. But on the whole, we are de-identifying samples. We are focusing on SARS-CoV-2 genome data. And that is how we got the first ethical waiver.
Okay, thank you, Piani. I hope that answered your question. And then we have one more. Have you tried using other genes like the N genes for your RT-PCR analyses?
We have some RT-PCR kits that also target the N gene. But those that missed the benchmark for validation of 90% sensitivity were not being used. We have a locally designed Skoda that is targeting ORF1AB. So we are not really using the N gene. Yeah, so Skoda also targeted the engine. Yeah, so we have Skoda out of those keys that is targeting the engine.
Okay, thank you. We also have let's see, 2 more questions that have just popped up. One is, I think it'll be good to understand some of the other prospects post-COVID. Would you mind sharing some insights into future projects of the lab?
Okay, okay. One important one was looking at associations of variants with severity of COVID-19. Then, of course, analyzing mutations in both non-VOC variants that we are getting in country to see which of these mutations within the spike gene, even though they are not VOC, that are highly under selection pressure in them. So another thing we are also doing is using bioinformatics to design primers for to be able to develop RT-PCR kit for rapid diagnosis of these lineages without sequencing them. So, with that we are also doing that. So, what we are also doing, if I may also share, by other team members, is looking at host genetic factors associated with COVID-19 severity. And one of them is looking at vitamin D receptor polymorphism and alteration in the expression of apoptotic genes to explain lymphopenia that we see in our patients. So these are some of the research activities that we are also doing aside sequencing at CRL.
Great, thank you. And one last question is, are there any ISO for the detection of SARS-CoV-2 by sequencing by analogy of ISO TS 5798-2022? So I assume it's regarding ISO compliance.
So thank you very much. So the lab which is actually in charge of processing pre-analytical, analytical processing of the samples collected is actually an ISO 15189 certified laboratory. And the sequencing lab currently hides under the umbrella of the quality management system of that laboratory at the moment. So yeah, thank you.
Okay, thank you, Dr. James. So just for the sake of time, I think we can end. So I'd also just like to make mention that we've worked with NIMA Lab to create an application note on the complete genome sequencing and identification of SARS-CoV-2 variants using the DNBSEQ-G50. So Prof did mention that in his presentation. And if any of you would like access, then you're more than welcome to reach out to us and we'll pass it on. So with that, I thank you all for your time. I thank you very much for the presenters today, and I wish you a good day. Thank you.